be a part of regarding natural gas exploitation in human health. And I first want to acknowledge that I am today on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and that Toronto is now home to many diverse First Nations, Inuit, and Métis people. I also want to apologize for everyone who were at my job talk in March 2020, as some of the content that I'm presenting today is similar, but hopefully you will still find that interesting. All right, perfect. So I just want to introduce myself briefly, as well as my education background. So I have a bachelor's degree in biology with a specialization in environmental health from the Université du Québec à Montréal. I graduated in 2010 and started at the same university a master's degree in biology, and more specifically in environmental toxicology. During my master, I used stable isotopes and GPS-based telemetry to understand the exposure to a class of contaminants called brominated flame retardants in ring-billed gulls. I then did my PhD in biology at INRS in human health. More specifically, I moved from environmental toxicology to toxicology. And during my PhD, I used and developed cellular bioassays to detect environmental contaminants that could act as endocrine disruptors. And those cellular bioassays were designed to mimic key endocrine processes during pregnancy and during the development of breast cancer. And it really gave me a solid foundation to better understand complex interactions between the environment and human health. In 2016, Marc-Henri Werner at the University of Montreal and myself instigated a pilot study on exposure to contaminants associated with hydraulic fracturing in northeastern British Columbia. And this led in 2017 to the start of my postdoctoral fellowship with Marc-Henri Werner on hydraulic fracturing and birth outcomes. And since I started working on hydraulic fracturing, I developed community-based research projects in close partnership with communities, First Nations, and also healthcare providers. So today I will talk about a specific research project, but I want to seize the opportunity to take a few minutes to present my research program. So since the beginning of my academic journey, different research projects contributed to establishing the research axis of my research program. So I worked on endocrine disruption by pesticides, like I mentioned, in breast cancer and fetal placental cellular models, to large-scale biomonitoring studies of emerging contaminants in Indigenous communities, and finally, numerous research projects on exposure to contaminants associated with fracking in northeastern British Columbia. My research program highlights community knowledge as a valuable source of information in order to explore the relationships between environmental factors and health. My lab pursues the following objectives. So first, to develop customized research strategies to identify potential environmental factors associated with health issues observed by communities, to investigate the prevalence and incidence of those health issues, to then develop or use cellular bioassays to study the biological mechanisms involved in the development or the progression of those health issues, and finally, to collaborate with regulatory agencies in order to hopefully contribute to adequate environmental and public health policies. So my research program focuses on 4 axes, all using a community-based research projects: hydraulic fracturing, emerging contaminants, pesticides, and industrial contaminants. And today, I want to present some of the research projects, and more specifically, one study that I have been working on that use data from multiple biological organization levels in the context of hydraulic fracturing. All right, so a natural gas is primarily composed of methane, and Canada is actually a large producer of natural gas. It's the 4th largest in the world. And natural gas has various applications, from heating to generating electricity, and it can also be used as an alternative fuel. Mainly, it's used in the residential, the commercial, the industrial, transportation, and the agricultural sectors. And to extract natural gas, industry uses a technique called hydraulic fracturing, or fracking for short. It involves many different phases that I will try to summarize here. So first, the drilling phase, where wells are drilled vertically, as you can see on the figure here, and then horizontally deep down in the rock formation. The second phase is the completion phase, where a perforating gun loaded with explosive charges is put inside the well, specifically in the horizontal part of the well. And when it is there, it will do its job, which is to create little holes in the casing of the wells, and that will create fractures in the rock formation. The fracking phase in itself is the injection of large volumes of what is called a fracking fluid, which is mainly water mixed with sand and different chemicals inside the well, deep down, all the way up to the horizontal part of the well. And the fracking fluid will come out of those little holes here and will get into the fractures, and that will free the trapped natural gas. Then there is the flow back phase, which is when the fracking fluid is pumped back to the surface of the well and is taken for disposal, or it can also be reused in another well. And then finally, the production phase of the well. This is where the natural gas is able to flow freely in the well and can then be stored in tanks and eventually transported in pipelines. So in Canada, hydraulic fracturing is mainly done in the northeastern part of British Columbia. That's the region you can see here on the map. British Columbia, the northeast part of that province, sits on the Mutney Rock Formation, which contributes to approximately 67% of the whole province's production of natural gas. And you can appreciate here the extent of the development, where all the little colored dots that you see are either individual wells or natural gas treatment facilities in the region. We know from previous scientific literature that there is potential contamination associated with that industry, namely by volatile organic compounds, and also trace and radioactive elements that are naturally occurring in the rock formations that are fracked and can then be demobilized from these rock formations and end up to the surface. So I now want to talk a bit about the state of the literature in terms of potential health impacts associated with fracking. And I first want to talk about what the toxicological data tells us. So in 2016, Elliott and colleagues published a systematic evaluation of around 1,000 chemicals identified in hydraulic fracturing fluids, and they screened those chemicals for their potential effects on reproduction and fetal development. And what they found is that for 76% of the identified chemicals, information on reproduction and developmental toxicity was lacking. On the remaining 24% of them, 43% of the chemicals were identified as being toxic for the fetal development, 40% of them for being toxic on reproduction, and 17% of them as being toxic for both reproduction and fetal development. And it's important to know that when Elliott and colleagues published that evaluation, there was no peer-reviewed exposure assessment studies in the context of fracking in the scientific literature. When we now look at the epidemiological data in terms of fracking and health, what we can see is that it's quite inconsistent. And I just want to give you one example of that. So the last time I checked, around 10 studies published in the scientific literature investigated the associations between proximity to hydraulic fracturing activity and birth outcomes. And 6 of those studies looked at birth weight as one of their outcomes. And while most of the studies did find an association between maternal proximity to fracking wells and a smaller birth weight, not all of those associations reached statistical significance. The studies were different on how they estimated exposure from fracking, how they measured the association between fracking activity and birth weight, and then how they reported those results. So for example, the studies done by Casey, Mackenzie, and Stacy all looked at the difference in mean birth weight between their highest and their lowest exposed groups. And while Casey found a significantly lower birth weight in the highest exposed group, as we can see here, Mackenzie found the contrary. Stacy also found a decreased birth weight in the highest exposed group, but they didn't reported the 95% confidence interval, which can make the interpretation of the results a little bit more challenging. So as you can see, there's not a lot of information regarding exposure to contaminants associated with fracking and the potential health impacts of that industry. However, the available toxicological data that we have tells us that reproduction and fetal development might be affected. And importantly, communities and some healthcare providers in Northeastern British Columbia have voiced concerns about the health impacts of this industry in their region, but they didn't receive support to answer their legitimate concerns. Because risks depend on both toxicity and exposure, we were first interested in conducting an exposure assessment pilot study in pregnant women. And I'm going to very briefly present you the results for some of the results of that pilot study. So we conducted this pilot study in 2016. The principal investigator was Marc-André Vermeer. We received funding from the University of Montreal, as well as the West Moberly First Nations. It was a community-based research project, so we worked closely with the Treaty 8 Tribal Association, the West Moberly and Saulteaux First Nations, and it involved the participation of 2 medical clinics where we did the recruitment of the pilot study. So our hypothesis was that exposure to contaminants known to be released during hydraulic fracturing activity would be more important in regions with substantial natural gas exploitation. So that's why we decided to measure gestational exposure to volatile organic compounds and trace elements in Northeastern British Columbia. Prenatal exposure to volatile organic compounds has been associated with a number of health outcomes, such as reduced birth weight, increased risk of childhood leukemia, birth defects, growth retardation, preterm birth, spontaneous abortion, and reduced fertility. And prenatal exposure to certain trace elements has also been associated with reduced birth weight, congenital malformation, miscarriage, and impaired neurodevelopment. So for this pilot study, we recruited 29 pregnant women in 2 cities in Northeastern British Columbia, Chetwynd and Dawson Creek. They, of course, answered a questionnaire on life habits, diet, etc. And we collected several samples, so 1 hair samples and multiple urine samples over 5 consecutive days. So we, first of all, measured metabolites of exposure to volatile organic compounds, one of those volatile organic compound being benzene. So we measured 2 urinary metabolites of benzene, SPMA and TTMA. So those are byproducts that come from the metabolism of benzene after we have been exposed to that chemical. We then compared the levels of those 2 benzene metabolites with what was measured in women from the general Canadian population as part of the Canadian Health Measures Survey, or CHMS. So CHMS collects information on Canadians' health and health habits, including exposure to environmental contaminants. So that really gives us a good idea of what the typical Canadian is exposed to in their day-to-day life. So what we can see when we look at the first benzene metabolite, SPMA, is that the median level in the pilot study participants was quite similar to women from the general Canadian population. However, when we look at the second benzene metabolite, TTMA, we can see that the median level in the participants from our pilot study was approximately 3 to 4 times higher than what was measured in the general Canadian population. We then compared levels of those benzene metabolites between regions of recruitment and also between Indigenous and non-Indigenous participants. And I'll show you an example of that. So the dotted line here represents the median TTMA level measured in women from the general Canadian population. The black line here represents a biological exposure index that is set as at 500 microgram per gram creatinine for TTMA. And what this means is that it's kind of an exposure guideline that is used as a warning of elevated benzene exposure in the workplace. So it's a guideline that is used in the occupational exposure setting. So, and the little squares that you see are individual levels of TTMA measured in the 29 participants from the pilot study. So what we can see, first of all, is that we had 5 participants that had TTMA level above the exposure guideline, and that the median level of TTMA in Indigenous participants seemed to be slightly higher than in non-Indigenous participants. As part of the pilot study, we also measured the suit of trace elements in both hair and urine samples. And we compared those levels to what was reported by a study done by Goulet and colleagues, who reported reference ranges of exposure to trace elements in healthy, non-occupationally exposed volunteers. And we did that because, unfortunately, when we did the pilot study, the Canadian Health Measure Survey did not include trace elements in hair, and that has changed since. So this is great, but at the time we didn't have those reference data. We did a more in-depth analysis for 4 trace elements that you see here: barium, aluminum, strontium, and manganese, because those 4 trace elements are naturally occurring in the Montney rock formation. And we hypothesized that they might be demobilized from the rock formation during fracking and reach the surface, therefore increasing exposure in human populations of that region. So the gray bars on the graph represent the median levels of those trace elements reported by Goulet and colleague as reference median levels, and the black bars represent what we measured in the pilot study participants. So we can see that for those 4 trace elements, the levels in the pilot study participants were quite higher than what was reported by Goulet and colleagues. Again, we compared levels between regions of recruitment and Indigenous and non-Indigenous participants. And for barium and strontium, we could see that levels of those 2 trace elements were higher in Indigenous participants compared to non-Indigenous participants. Here you can see the levels in hair, but we had similar results for urine as well. So the pilot study had several limitations. First of all, we had a very small number of participants. Another limitation we had was because the TTMA, which is a metabolite of benzene exposure, is not 100% specific to benzene. You can have TTMA in your urine samples that come from the metabolism of something totally different than benzene, which is sorbic acid. It's a quite common food preservative. Another limitation we had was that we didn't measure VOCs and trace elements in the participants' environment, and that the reference population we had for trace elements was not the best reference population because of a lack of data in the Canadian Health Measure Survey. But we can conclude still with the pilot study that we did observe higher level of 1-benzene metabolite, namely TTMA, and that it was even higher in Indigenous participants when we compared to the Canadian general population. We could also see that for 4 trace elements in particular, barium, aluminum, strontium, and manganese, the levels in the pilot study participants were quite higher compared to reference medians reported by Goulet and colleagues, and that for barium and strontium, the levels were also higher in Indigenous participants compared to non-Indigenous participants. So the pilot study was the catalyst of 2 major studies that are ongoing and part of my research program, including an epidemiological study on proximity and density of hydraulic fracturing wells and birth outcomes in northeastern British Columbia. So this work was started, like Michelle said at the beginning, during my postdoctoral fellowship, and it was also a community-based research project. So we worked closely with the Treaty 8 Travel Association. So based on previous epidemiological literature and toxicological data regarding contaminants associated with fracking, we hypothesized that proximity to fracking activity could contribute to poorer birth outcomes. And that's why we wanted to evaluate the associations between maternal residential proximity to fracking and birth outcomes using birth records data from the largest hospital in that region, which is the Fort St. John Hospital, over a 10-year period. So the first step was to collect the birth records data from the public health agency over there, which is called Northern Health. Then to estimate maternal exposure during your pregnancy with the IDW method. So I will explain that in the next slide. And then to measure the associations between exposure metrics and birth outcomes. So in epidemiological studies, we often work with data sets and we don't necessarily have access to biological samples that we can use to measure the exposure to contaminants of interest. We have to rely on methods that allow us to estimate the exposure rather than measuring it. And that's the case for the IDW method, which stands for inverse distance weighting method. And it's based on the density and proximity of fracking wells to a point of interest. It could be a residential address, or in our case, we use the postal code centroid that was associated with each birth. And the reason for that is simple. It's just we didn't have access to the physical address of all the mothers who gave birth at the Fort St. John Hospital. We had access to their 6-digit postal code for confidentiality reasons. So we first obtained the oil and gas data from the British Columbia Oil and Gas Commission. They maintain a provincial database of oil and gas wells location and some characteristics. The IDW metrics are generated by using buffers around the postal code centroid. So there you go, we have our buffer. We then count the number of wells that are present within that buffer zone. So that's the density part of the IDW metric. We then measure the distance between each of those wells to the point of interest, so the postal code centroid in that case. And then we combine that information in the IDW formula, which will basically put more weight to the wells that are the closest to the residence or the closest to the postal code centroid. And that kind of gives us, a number that is in relation to the number of wells around the residence and the distances of those wells to the residence. So for each birth, we conducted 3 IDW measurements using 3 different buffer zones. So a 2.5, a 5, and a 10-kilometer buffer zone around the postal code centroid. And we then categorize the IDW metrics into quartiles of exposure. And the first quartile was used as our reference group. So the first quartile would be the mothers that were the least exposed to fracking activity during their pregnancy. So just a few characteristics of that birth cohort. 58% of the mothers have had previous pregnancies. And so those characteristics are for around 6,000 births that occurred at the Fort St. John Hospital between 2007 and 2016. 12% of them reported smoking during their pregnancy. We didn't have the information on the gestational age of around 1,000 mothers, but on the remaining births that we did have that information, 10% of the infants were small for their gestational age or SGA, and 4% of them were born prematurely. So I'll just show you one result of that study regarding birth weight. So we evaluated the associations between the well density and proximity metrics, so the IDW metrics, and birth weight. And to do that, we used multiple regression analysis, and we corrected the models for a number of important covariables, such as parity, infant sex assigned at birth, mother's age, and smoking. So on the figure here, you can see the adjusted beta, so that's the central point here, with their respective 95% confidence interval for each of the 3 buffer zones, so the 10, the 5, and the 2.5 kilometer buffer zone, and for also each of the quartile of exposure, so the 2nd, the 3rd, and the 4th quartile of exposure. The beta is the degree of change in the outcome variable, so in this case, birth weight, for every unit of change in the predictor variable. So in this case, the well density and proximity metrics. So a beta of 0 means that there's no relationship whatsoever between your predictor and the outcome variable, while a negative beta would mean that there's an increase in exposure would yield to a decrease in birth weight, and a positive beta would mean that an increase in exposure would yield to an increase in birth weight. So after adjusting for the covariables, we found a negative associations that did not match the classic dose-response relationship. So for example, women in the 2nd quartile of the 10-kilometer buffer and women in the 2nd and 3rd quartiles of the 5-kilometer buffer of well density and proximity metrics had babies with birth weights between 41 and 47 grams lower than the reference quartile. So in this epidemiological study, we also had limitations. The IDW is based on the postal code centroid and not the physical address, which can lead to misclassification in exposure. And it's also not considering the different phases of production, the ones that I presented earlier in the talk, which we know can influence the type and also the concentration of contaminants emitted. We also didn't have access to potentially important covariables such as maternal education, family income, or ethnicity. But it was the first epidemiological study in Canada on hydraulic fracturing and health. We did see potential associations with reduced birth weight and also increased odds of preterm birth, but I didn't present the results today for the sake of time. We didn't see any association with small for gestational age or head circumference, and hopefully we will be able to eventually explore the mechanisms that can potentially explain the weird dose-response relationship that we see, meaning that the more striking effects that we measured were not seen at in the highest exposed group, but rather the medium exposed group. So right now we're looking at, we almost done with the manuscript regarding cardiovascular and respiratory outcomes, and I'll dig into the endocrine related birth defects. And we're also working on another epidemiological study on hydraulic fracturing and asthma exacerbation in the same region with colleagues from UBC and SFU. All right, so the 2016 pilot study, if you remember, allowed us to observe high levels of one benzene metabolite and several trace elements in urine and hair samples of pregnant women from northeastern British Columbia. And we know that these compounds can transfer through the placenta and reach the fetus. So in a bigger, larger biomonitoring initiative, we want to confirm or disconfirm the results from that pilot study. And we also know that endocrine disrupting activities have been observed for a variety of chemicals associated with fracking. So we also aim to investigate the endocrine disrupting potential of low environmentally relevant concentrations of contaminants associated with fracking. And that leads me to present you our current study, which is called Experiva for exposures in the Peace River Valley. So Experiva is funded through a project grant with CIHR. There are both two PIs, Marc-André Werner at University of Montreal and myself. We also put in place an oversight committee whose mission is to evaluate all the aspects of the research project prior to their use. As you can see, we have a large research team, and it is also a community-based research project. So we work closely with the same community partners, Treaty 8 Tribal Association, West Moberly, and Saulteaux First Nation. So Experiva is really a central project of my research program. It is a transdisciplinary project that combines exposure assessment, toxicological, and sociological approaches. Given the limitations of the pilot study, we built Experiva around those limitations to try to address them as best as we could. So the first objective of Experiva is to measure the exposure to volatile organic compounds and trace elements, but this time in a larger cohort of pregnant women in northeastern British Columbia. The second objective is to assess endocrine disruption of those environmentally relevant concentrations of volatile organic compounds and trace elements. And we are using cellular models of fetal development to do that. The third objective is to explore the associations between density and proximity of fracking wells and exposure levels of those 85 participants, as well as between their exposure levels and birth outcomes. And finally, to explore social inequities related to exposure to contaminants associated with fracking. So the recruitment of Experiva was conducted in northeastern British Columbia from May to August 2019. It involved the participation of 3 medical clinics and a brand new midwifery clinic. It was also done in partnership with the Treaty 8 Tribal Association, West Moberly and Saulteaux First Nations, who advertised the project to their members. There are approximately 800 births every year at the Fort St. John and Dawson Creek hospitals. So we estimated that we recruited approximately 33% of the pregnant women in the region at that time. And 90% of the pregnant women that were met agreed to participate in the study. So we were quite happy with the success of the recruitment. So today I will present you some preliminary results regarding the objectives 1 and 3 of Experiva. So for the first objective on exposure assessment, we are using a multimedia approach. So we first have environmental monitoring. So we measured volatile organic compounds in indoor air and tap water of the participants. And in tap water, we are also measuring trace elements. And the participants provided a lot of biological samples, so multiple urine samples, also nails and hair. For nails and hair, we are using those samples to measure trace elements, and it gives us an idea of their longer-term exposure to trace elements. And in urine, we measured also trace elements, and we are currently measuring metabolites of volatile organic compounds. We then compare levels of those contaminants to the general Canadian population. And an important part of that project is to present the results, obviously, to the participants and the community partners. And that was done remotely last summer. For the third objective on the associations between density and proximity of fracking wells and exposure levels, we used a similar IDW approach that previously described in our epidemiological study. So first, we calculated the density of wells around the participants' residence. So just the number of wells using the three same buffer zones. And we also calculated the IDW metrics that takes into consideration the density and the proximity of the wells. And we used the same 3 buffer zones as well as a no buffer zone, so accounting for all wells present in the province. We then measured the associations between those exposure metrics, so density of wells and well density and proximity, with the actual levels of exposure in the 85 participants for the Experiva study. So specifically for volatile organic compounds levels in indoor air and tap water. And to do that, we use multiple linear regression models. All right, so a few characteristics of Experiva. The median age of the participant was 29 years old. 20% of them identified as Indigenous. The median gestational age of the participants was 23 weeks pregnant. 68% of them have had previous pregnancies before this one. 8% of them reported smoking during their pregnancy, and 23% of them reported being exposed to secondhand smoke during their pregnancy. 22% of them had an attached garage to their residence, and 51% of them had city water for their residential water. So smoking, exposure to secondhand smoke, and also having an attached garage can all contribute to the levels of volatile organic compounds measured indoor. And also the source of water can contribute to the levels of exposure to different contaminants in the tap water sample. So that's why we collect that information in the questionnaires. I thought it would be interesting to show you what it looks like in terms of density of wells and residences of the participants. So during Experiva, we identified 29,588 hydraulic fracturing wells in the region at the time of the recruitment. And so those are the kind of map this is the kind of map that we generate. And this is just all the wells that were present at the time of the recruitment for Experiva. The second map here, the wells are shown in the pale green, and the purple dots that you see represent the residents of the Experiva study participants. And this is just a close-up of the region. So you can really appreciate how the wells can be close to the participants, to anyone that lives in Northeastern British Columbia, really. It's important to note, however, that not all these wells are necessarily active. Some may be abandoned, some may be decommissioned, inactive, or in different phases of production. But it's just to give you a visual idea of what it looks like. In Experiva, the median number of wells around the participants' residence in a buffer zone of 10 kilometers was 90 wells, and the maximum was 346 wells. So, that's quite significant. So here you can see the median levels of multiple volatile organic compounds measured in the indoor air samples of the Experiva study participants in the blue-green bars. and the median indoor air levels of those same volatile organic compounds reported in the Canadian Health Measures Survey, or CHMS, in the orange-yellow bars. So again, CHMS provides nationally representative levels of VOCs in indoor air. So of the 47 VOCs that we analyzed in indoor air samples of the Experiva participants, 40 of them were detected in more than half of the participants and also had nationally representative levels provided by CHMS. For 27 out of those 40 VOCs in residential air, median levels in the Experiva study were between 1.3 to 3 times higher than the national median levels. And in particular, for the VOCs that you now see identified by the red boxes, the proportion of participants from the Experiva study with indoor air levels of those VOCs over the 95th percentile reported by CHMS ranged between 10 and 28%. So the CHMS 95th percentile for the indoor air level of a specific VOC means that 5% of the time, indoor air levels of this VOC will be higher than that particular limit that is called the 95th percentile. So if the exposure to VOC in the Experiva study was similar to the general Canadian population, we would also see around 5% of the participants with indoor air VOC levels above the 95th percentile in the general population. What happens is that for a good number of VOCs, we have more than 5% of the participants with indoor air levels above the CHMS 95th percentile. And although those specific VOCs can have many different sources of exposure, most of them are either used in hydraulic fracturings or have been measured in hydraulic fracturing wastewaters. We also analyze a suite of VOCs in tap water. And over the 40 VOC that we analyzed in Experiva, we could only detect 4 VOCs, and only 2 had nationally representative levels in the general Canadian population. And that's the 2 you can see here, bromodichloromethane and total trialomethanes. So what we can see is that for bromodichloromethane, the median level in Experiva was higher than the general Canadian population, but we didn't have any participant with bromodichloromethane in tap water above the 95th percentile in the general Canadian population. However, for trialomethanes, the situation is different. So not only the median level in Experiva is higher than in the general Canadian population, but we also have 60% of Experiva study participants with trialomethane levels in tap water above the CHMS 95th percentile. So bromodichloromethane count as part of the total trialomethane measurement. They are both common disinfection byproducts in tap water. However, it is known that wastewaters generated by fracking contain high concentration of total dissolved solids, which is believed can lead to increased concentration of trialomethanes at drinking water treatment facilities that disinfect their water using chlorination. To finish the talk, I want to present 2 last results. So regarding associations between density and proximity of wells and actual VOC measurement. So many VOCs measured in indoor air samples in Experiva were strongly correlated with each other. So we selected just a few VOCs to conduct those analyses and VOCs that were not correlated with each other. So I present you results for 3 of those VOCs, acetone, 2-methyl-2-propanol, and chloroform. So after adjusting for confounders, we found a positive association between the 10-kilometer buffer well density metric, so here, and acetone and chloroform indoor air levels. We also found similar association here when we use the all wells IDW metrics. It's important to note that the indigenous status in those models contributed significantly to the levels of acetone and chloroform in indoor air. And this is further illustrated by the fact that indoor air levels of those 2 VOCs were higher residences of indigenous participants compared to non-indigenous participants. In those association models, we also conducted sensitivity analysis to account for other sources of exposure to VOCs other than fracking. So for example, we included the use of nail polish remover in the week of the sampling, as well as if the participants were storing paints and solvents in their home or in their garage. And for the chloroform model, we also included the source of residential water, as volatile organic compound present in tap water can evaporate and then be present in indoor air. And in all of those sensitivity analysis that we conducted, they still supported a significant association between the density and the proximity of fracking wells and indoor air concentrations of acetone and chloroform. We also did similar models for trialomethanes in tap water. And again, we also saw that trialomethanes were significantly higher in residences of Indigenous participants compared to non-Indigenous participants, as well as in participants whose source of residential water was municipal, which makes sense because this is chlorinated water. So after adjusting for confounders, we found positive association between the 10 and 5 kilometer buffers density metrics and trialomethane measurement in tap water. And we found similar association when using the IDW metrics for 10 and 5 kilometer buffer zones. So what's next here will be to compare levels of trace elements in tap water with what is measured in the general Canadian population, and to analyze the levels of exposure to VOCs and trace elements in the biological samples of our participants. So we're currently doing those analyses. Another step that we are currently doing is the second objective regarding the endocrine disrupting potential of volatile organic compounds. So we are using a fetal-placental co-culture model that I used during PhD that really mimics well the interaction between a fetus and a placenta. So the co-culture model can de novo synthesize all the hormones that are produced during a real pregnancy. And we will expose the co-culture model to the concentrations of volatile organic compounds and trace elements similar to what was measured in the participants of the Experiva study. And we will look at a number of outcomes in the co-culture, such as hormone levels and gene expression and protein activity and that kind of good stuff. Another step that we are taking is working on estimating the exposure of the Experiva study participant to other chemical and non-chemical stressors, such as particulate matter and perceived environment quality. We will also quantify over the summer the levels of multiple oxidative and chronic stress biomarkers in the urine samples of the participants to investigate the association with chemical and non-chemical stressors in the Experiva study. And this is really interesting in the context of that study because oxidative and chronic stress have been identified as important mechanism implicated in the etiology of negative birth outcomes. All right, so to conclude, we do have some epidemiological literature that suggests risk for poorer birth outcomes, but effects do not always reach statistical significance. And we hope to continue to conduct epidemiological studies in Northeastern British Columbia. I feel like it's deeply needed. We are still facing an important lack of data, specifically on exposure assessment and also mechanisms of toxicity. So till this date, the 2 papers for the pilot study regarding exposure to volatile organic compounds and trace elements are still the only 2 papers published in the literature that specifically looked at exposure levels in the context of fracking. So definitely not enough data on that. In Experiva, we do have some preliminary evidence of higher exposure levels to volatile organic compounds, especially in Indigenous participants compared to the general Canadian population. And we also have preliminary evidence that density and proximity to fracking wells contribute to the level of exposure to those of some of those volatile organic compounds. To conclude, my research interests is, I hope you could see, at the nexus of toxicology, molecular biology, community-based research, environmental epidemiology and public health. And our current research projects in the context of fracking are aligned with the objectives of my research program, From Bench to Communities, which is to develop transdisciplinary research projects in partnership with communities that live in a changing environment, and to assess the impacts of those anthropogenic pressures on health by combining information from multiple levels of biological organization. And that's it for me. Thank you for your attention. I hope this was interesting.